So today's topic is glutamate. So glutamate are the excitatory type of neurotransmitter, the principal excitatory neurotransmitter in CNS, okay, and they are stored in the neuronal cell membrane. So glutamate comes in the CNS with the help of, it is present in the CNS in glial cells as well as in the Krebs cycle. These are responsible for neural communication, memory formation, learning and regulation. So these are their function. In the CNS, we will see the glutamate synthesis. Now, glutamine is converted into glutamate with the help of the glutaminase enzyme. Okay, glutamate is stored in the synaptic vesicle as any other neurotransmitter is stored. From synaptic vesicle, glutamate is released by the process of exocytosis, which is a calcium dependent process. Any neurotransmitter which is stored in the synaptic vesicle, it is released by the process of exocytosis, which is a calcium dependent process. Next is, we will see the diagrammatic representation of glutamate synthesis. So neurons, glutamine is converted into glutamate with the help of the enzyme glutaminase, okay, and then it is released with the help of the exocytosis and it enters into the postsynaptic junction, okay. So glutamate receptors, we will see glutamate receptors are the synaptic receptors located primarily on the membrane of neuronal cells. Okay, these are the synaptic receptors. So glutamate, or you can call it as glutamic acid, it is present in the body, but mainly it is particularly most abundantly present in the nervous system, okay, or in the CNS, in the human brain. Okay, it has main activity as a excitatory neurotransmitter. It is precursor for GABA, okay, as we have seen in GABA, glutamate then converted into GABA. So glycine, then glutamate, then GABA. So it is a precursor for GABA, the brain main inhibitory neurotransmitter. Okay. So this is an excitatory neurotransmitter. Glutamate receptors are responsible for the glutamate mediated postsynaptic excitation of neuronal cells and are important for the functions which are neuronal communication, memory formation, learning and regulation. Okay, so glutamate receptor are of two types, inotropic and metabotropic. So if they are metabotropic, they are regulated with the help of the second messenger. Therefore, they are G protein coupled receptors, and inotropic are ligand gated cation channels. Okay, so inotropic receptors are of three types: kinase, AMPA, NMDA. Okay, and metabotropic receptors are subdivided into three groups: group 1, group 2, group 3. Okay. First, we will see the inotropic receptor. Inotropic receptors are ligand gated, non-selective cation channel. Okay, it allows the flow of every cation, which is potassium, sodium, and sometimes calcium, in response to the glutamate binding. Here we can see the ligand gated ion channel. Neurotransmitter bind the when the neurotransmitter bind, the ion from the outside of the cell enters into the across the membrane, enters across the membrane. Okay, when the neurotransmitter binds. So these ions are mostly cations. Inotropic glutamate receptor are of three types: NMDA, AMPA, and kinase receptors. Okay, so first we will see the NMDA type of receptor. The NMDA receptor, which is also known as N-methyl D-aspartate. Okay, so the NMDA receptor is a glutamate type of receptor, which is important, which plays an important role in potentiating learning and memory. Okay, so NMDAR is a specific type of inotropic glutamate receptor. It is an inotropic receptor. NMDA is the name of the selective agonist that binds to the NMDA receptor. So NMDA actually binds on the NMDAR. Okay, so NMDA, N-methyl D-aspartate, acts as an agonist. Okay, but it does not bind to other glutamate receptors or their subtypes. It only binds to the NMDAR receptor. Okay, calcium flux through the NMDAR is thought to be critical in synaptic plasticity. The role is here NMDA receptors, the ion which influxes is calcium, and it is mostly thought to be critical for the synaptic plasticity. Okay, a cellular mechanism for learning and memory. So NMDA agonist, and glycine is the antagonist in the NMDA receptor. Okay. Now NMDA receptor agonist, these are the examples of agonists like L-aspartate, quinolylate, homocysteine, D-serine, ACPL, L-alanine, okay, and partial agonist, N-methyl D-aspartic acid, 3,5-dibromo-L-phenylalamine. So these are the agonists. NMDA receptor antagonist, amantadine, ketamine. Ketamine is an euthanasia or anesthesia agent, okay. Methotrexamine, nitrous oxide, okay, nitrous oxide is a vasodilator. Lead, xenon, okay, these are the examples of NMDA receptor antagonist. Now we will see the next receptor type, which is AMPA receptor, okay. So the alpha amino 3 hydroxy 5 methyl 4 isoxazole propionic acid receptor, okay. So that is AMPA, okay, so also known as AMPA receptor, AMPAR, okay. So this receptor is non-NMDA type of inotropic transmembrane receptor for glutamate, okay, that mediates the fast synaptic transmission in the CNS. So what does AMPAR do? So they are mostly found in the parts of the brain and most commonly found receptor in the nervous system. They are most commonly found, okay, in the CNS. The agonists are 5-fluorovilladilin, AMPA, okay, domoic acid. Antagonists are ethanol, priampanil, okay, tizampanil. So these are the antagonists. So how does they act at the axonal membrane? Glutamate is stored in the synaptic vesicle. They transfer to the synaptic cleft via exocytosis. Now here the ions like sodium, magnesium, they bind to the NMDA receptor, okay, and AMPA receptor as well, and hence they can easily transfer inside the postsynaptic membrane, okay. So here with the high presynaptic activity of most of the AMPA receptor are activated and the excitatory postsynaptic potential is strong. So what they do is that they potentiate the activity of NMDA receptor, okay. So the strong EPSP, okay, lifts the magnesium blocks of the NMDA receptor. The calcium signal ultimately leads to the synaptic strengthening. So synaptic strengthening is the function of the AMPA receptor. As such, their activity are not that fast. So what they do is they potentiate the activity of the NMDA receptor, okay. So they increases the EPSP, okay, and make it strong. That's why they then ultimately lead to the synaptic strengthening. So calcium, it acts as an important secondary messenger, okay, and activates the intracellular cascade. Now calcium binds to the calmodulin protein. You know that when calcium enters, what happens? It binds to the calmodulin protein, okay, which activates the protein kinases like CAM kinase, okay. So then the vasoconstriction or constriction occur, okay. So that is the function of the calcium. Now CAM kinase affect AMPA receptors in two ways. What happens? It phosphorylates the AMPA receptor already present in the dendritic, means obviously the phosphorylation will takes place, okay. It increases the conductance of the sodium ion inside the cell, obviously promotes the intracellular AMPA receptors to move to the membrane, making more receptor available to stimulate the spine. Thus the long term potentiation of the AMPA receptor is done. The next type of receptor we will see is the kainate receptor. Kainate receptor or KARs are inotropic receptor that respond to the neurotransmitter glutamate. KAR are less understood than AMPA and NMDA. As such, their activity are unknown. Okay, so physiological studies have identified both post and presynaptic roles for kainate receptor. Okay, so presynaptic activity of kainate receptor is to facilitate or reduce the neurotransmission depending upon where they are in brain, and postsynaptic kainate receptor can indirectly mediate the excitatory transmission. Okay, so as said, they don't have the activity, but if they are present in the presynaptic membrane, they will reduce or they will control the neurotransmission, and if they are present in the postsynaptic, they will mediate the excitatory transmission. They will facilitate, they will potentiate. So the antagonist example is ethanol, kynureninic acid, etc. Okay, so physiological and pathological roles. AMPA receptors, they mediate the most fast EPSPs in the CNS. Kinases, they are regulation of neuronal excitability, epilepsy, excitotoxicity, and pain. So because they are excitatory neurotransmitter, they can cause the excitotoxicity, okay, which will lead to epilepsy, schizophrenia, etc. So NMDA receptors also, they mediate the fast EPSPs in the CNS. They act via anesthesia action, learning and memory, okay, developmental plasticity, epilepsy, schizophrenia, excitotoxicity, okay. The next part of the receptor is metabotropic glutamate receptor. Metabotropic glutamate receptor are the G protein coupled receptor activated by the glutamate, the major excitatory neurotransmitter in the CNS. So neurotransmitter, if they are G protein coupled, okay, bind receptors. So neurotransmitter binds to the receptor. Okay, now here we know that in the G protein coupled receptor, they will either when the neurotransmitter binds to the receptor, the alpha subunit will detach from the G protein coupled receptor, G protein, and then they will enter into the effector protein, okay, and thus the intracellular messenger will activate the glutamate protein, okay, glutamate receptor, whichever receptor it may be, metabotropic receptor. So and thus the ion will flow across the membrane, ion channel, it will open the ion channel, okay. So G protein will be activated, G protein subunit or intracellular messenger modulate to the ion channel, and the ion channel thus opens up and the ions enter inside the membrane. So G protein coupled receptor, as you know, they contains 3 units, which is alpha, beta, and gamma. 2 primary signaling cascade, which we can also call the secondary messenger CAMP pathway or phosphatidyl inositol pathway. The pathway activation depends upon the alpha subunit. Okay, you know that S unit, I, the types of G protein S, I, Q. So okay, and GDP bound to the alpha when they are inactive. It gets converted into GTP afterwards when they are activated. So G protein coupled receptor, when a ligand binds the receptor, okay, when a ligand binds the receptor, obviously changes the conformation, allowing the G protein to be activated. Okay, G protein dissociate from the receptor, then the subunits to each other. CAMP pathway, as you know that G alpha S binds to the adenyl cyclase and stimulate the CAMP synthesis from ATP. Okay, and GI they inhibit. Okay, adenyl cyclase pathway, will see that ligand bind to the receptor and then the ATP is converted into CAMP with the help of a secondary messenger, which is adenyl cyclase. Okay, and same is for the inositol. So this is the glutamate receptor overview: NMDA receptor, AMPA receptor, kinase receptor, and metabotropic receptors. Okay, here agonist, antagonist, modulators, channel blockers are given, the mechanism is given, the location and their functions are given. Thank you.